Hola, buenos días a todos y todas por acompañarnos de nuevo a un seminario del Instituto de Biología de la Universidad Nacional Autónoma de México. Hoy nos acompaña el doctor Milan Janda. Hoy es el último seminario del semestre. Estaremos reanudando los seminarios a finales de agosto, o a finales de julio, perdón. Voy a dar un par de minutos para que entren más personas y luego presentaré al doctor Janda. Okay. Okay. Ya tenemos audiencia. Ok, entonces les voy a hablar un poquito sobre el doctor Janda. El doctor Milan Janda realizó sus estudios de posgrado en la Universidad de South Bohemia, en la República Checa. Dentro de su producción intelectual están más de 30 publicaciones en revistas indexadas y miles de citaciones. Milan actualmente trabaja como investigador con ACID en el Laboratorio Nacional de Análisis y Síntesis Ecológica. Su laboratorio incluye múltiples estudiantes, tanto de licenciatura como de posgrado, y sus intereses en su laboratorio son la ecología molecular, la diversidad tropical, las interacciones entre especies y la filogografía, biogeografía, genética de poblaciones, evolución y sistemática de las hormigas. Hoy el doctor Janda nos va a hablar de la ecología y evolución de las hormigas en islas. Su presentación va a ser en inglés, sin embargo, las preguntas pueden ser en español o en inglés, como ustedes prefieran. Milan nos va a hablar por aproximadamente unos 45 minutos y luego tenemos espacio para sus preguntas a Milan. Entonces, Milan, bienvenido. Muchas gracias por aceptar nuestra invitación. You can take it away.
Muchas gracias, Ivonne, por la introducción y por la invitación a participar en este ciclo de seminarios. Es un honor participar y estoy muy feliz que el Instituto de Biología fue siguiendo con las charlas durante los últimos meses. Y entonces, disculpa por platicar en inglés, creo que puede ser un poco más preciso. Pero si vamos a tener algunas preguntas o discusión, podemos después regresar a español.
Está perfecto, no te preocupes. Nos vemos en un ratito.
Our group is based at Laboratorio Nacional de Análisis y Síntesis Ecológica, which brings together a diverse group of researchers. They are based in ENES-UNAM and focusing on various aspects of ecology. One of the important topics which we are studying there are the interactions between the plants and pollinators. There are groups studying the population genetic and conservation genetic of plants. There are groups dedicated to the evolutionary immunology and so analysis of the spatial models and remote sensing and many others. In my laboratory, we are using insects and ants in particular as models to study ecological and evolutionary processes which generate biological diversity. We are often using phylogenetic approaches to study ecological history of certain species. We are interested in island ecosystems. We are also interested in interactions among the ants and plants. And also important line of our research in Mexico is development of the resources for monitoring and prevention of invasive species. So what's so interesting about the islands? So they are considered by biologists as natural laboratories because of their discrete nature, can facilitate reconstructions of diverse evolution and biogeography histories of resident flora and fauna. We can consider them as semi-independent replicas where we can study the same evolutionary and ecological processes in slightly different settings. Although they represent only a small portion of the land area, they contribute a lot into the global biodiversity and also to the cultural diversity. Unfortunately, over the last 500 years, islands were also where majority of the documented species extinction occurred. So these are very endangered ecosystems in big need of conservation and studies. So there are good model systems for investigation of the fundamental biogeographic processes. And there are several principal processes which are influencing the biodiversity on the given islands. Today, we will be talking mostly about the topics of species-area relationship, about domains of MacArthur-Wilson dynamics. We will talk about colonization and evolution. I will try to show you how certain ecological traits facilitate local diversification or colonization of the new islands and areas in ENES. Melanesia and South Pacific have always attracted biologists. It is because of its complex geographic settings. It's among the most diverse region on the planet. And so it's not a coincidence that local flora and fauna was essential in development in some of the important biological theories over the last centuries. And it's not only the island biogeography models by MacArthur and Wilson, but the visit in Western Melanesia was very important for Alfred Russel Wallace, and also the different groups from Melanesia and South Pacific were important for other biogeographical researchers like Ernest Mayr or Jared Diamond. So what makes ants great models for biodiversity studies? It's, as many of you have experienced, they are super abundant and omnipresent in most of terrestrial ecosystem in tropics and subtropics. They are not only important predators, but also herbivores, detritivores. They enter many important mutualistic interactions with plants and other animals. We also think that their biodiversity patterns are somewhat representative of other invertebrate groups. And because of their colonial nature, they are important models for the social evolution. I would also say that it's quite a democratic group to study, because over the last three decades, there were many online resources developed for this group. So today it is possible to access all the taxonomic literature, most of the photographs of the type specimens. There are great distributional databases and databases of ecological data, which allow to use this group as a great biodiversity as the model for biodiversity studies, which is easily accessible. More than 60 years ago, E.O. Wilson visited New Guinea and Melanesia, and his collections and work of other colleagues had important impact on taxonomic and ecological studies of ants in the region. But also, these were essential for articulating the theory of the island biogeography. Using the ants from Melanesia and South Pacific, they constructed the first species area curves, also with the data from birds and the plants, and then later used these data for articulating the island biogeography, equilibrium theory of island biogeography. Just a brief recapitulation. So this is a textbook example. What is it about? So basically, the number of species on the island is the function of the immigration rate and the extinction rate, and these curves have different slopes depending whether the area and the island is small or large, or whether the island is far or near from the source, from the mainland. And of course, this theory has been simple, has been hugely successful, allowed us to test many questions, and not only on the island ecosystems. In addition to the island biogeography theory, Wilson also developed a series of hypotheses which became known as a Taksim Cycle, which is a non-equilibrium model which incorporates non-neutral processes, unlike the previous theory. And basically, it says that the Taksim Cycle narrative is that the dispersal and ecological adaptation interact with the competition, with the range contractions and extinctions to explain observed species distribution. So he hypothesized that the species can go through 3 different stages. First is the stage 1, where the species expand their geographical ranges across archipelagos, often aided by some ecological release, and shifts to occupy marginal or secondary habitats. In stage 2, there is an extinction and population differentiation, which leaves patchy distributions of the species across archipelagos. And then in stage 3, the island endemics can evolve the potential either further radiate within the islands or reinitiate the Taksim cycle by changing the habitat preferences, entering stage 1 and expanding to other areas. He also made some pretty exact predictions for some of the ant species in Melanesia, where he classified them into different stages and then observed where they live, and that basically created a framework which we can now test with the phylogenetic approaches. However, as the new data on ants were gradually accumulating from the region, we were realizing that the data used for the development of the theory of island biogeography were often partial and far from complete. Also, some of the predictions made by Wilson and colleagues didn't seem to fit some of the new data. And it's not surprising because their studies were based on very limited sampling. It turns out gradually that faunas of many of these islands are much more diverse. And also development of the systematic studies changed a lot of species limits and also brought more new information about the phylogenetic history of many of these lineages. So it became more apparent that these original data are due for some reanalysis and reassessment. So some of these things which E.O. Wilson was proposing about distribution of ants in Melanesia and Pacific were, for example, that there was no apparent effect of isolation on island species richness across Melanesia. Of course, although the isolation limits colonization of those lineages, he argued that this is compensated by greater rates of local speciation. And so basically, then the diversity remains the same. Tyler and Wilson were also arguing that the lack of endemics east of Samoa is the evidence that these species didn't reach those areas on their own, but they were brought by humans in the last 400 years. Although we know that Melanesians and Polynesians are avid travelers, and there were a lot of historical connections among these areas, it also seemed that faunas of some of these islands were quite large, although they didn't contain any endemics. These were species which came from the region, so it looked slightly suspicious to consider that all these species were arrived with humans. Although, of course, it is well known that the human colonization of these places changed considerably the ecosystems on many of these islands. So, with our colleagues, led by Evan Economo, we assembled the data for over 200 islands, forming several archipelagos. So here, just to orient yourself, so that's New Guinea, Micronesia, Melanesia, and then South Pacific Islands. And so we looked at the data, we analyzed the data about and looked at the species area relationship. What we find out is that total ant richness is strongly correlated with the area and only secondarily correlated with the isolation. Yeah? So here we have the area and then the isolation. Yeah? And then depending which part of the fauna we consider, then these correlation coefficients are stronger or weaker. So what was interesting is that if we consider the total fauna of those islands, then there is a strong species area effect and much more modest isolation effect on the total species. So this is the variance portion of the variance explained by different parts. So this is the isolation and the area. And if we consider the total fauna of those islands, the area explains majority of the variance. However, if we only consider the colonizing lineages, that means lineages which originated in the region but are not and arrived to the islands on their own, or the total natives, which also include these species plus local endemics, then there is a more stronger effect of isolation rather than area. So it turned out that after this analysis, that it appears that some of these native faunas are smaller than expected, which is due to the isolation. And the endemic species are present only in large archipelagos of low and intermediate isolation. So it seems that endemics and the ants to create endemic species need relatively large size of the islands and ideally large size of archipelago, which is not very isolated. They actually it seems that they need somewhat larger area than most of other insects in order to create endemic species. So we also saw that archipelagos which can be considered empty, that means their native fauna was well below the relationship, also receive more exotic species. So these human introductions compensated for the lower diversity and were basically tightening the species area curve. And so the less diverse the local fauna was, then there was more exotic species established on the island. So, of course, all this can be done different ways, and important part of our research is to go to the field and survey the local fauna. So now I'm just going to talk a little bit about our field work, mostly just to show you some photographs. And since 15 years ago, we've been studying ants in New Guinea. I basically started there during my master studies, and as a part of the big research group, which is focusing on interactions between the insects and the plants, under the umbrella of the New Guinea Binatang Research Center, led by Wojciech Nowotny from Czech Academy of Science. So this has been one of the most productive organizations in biology in Melanesia for the last 20 years, specializing on these large-scale ecological studies and also whole ecosystem experimental manipulations, for example. So we were lucky enough to visit remote areas of the islands and survey the fauna across different mostly lowland areas, using all kinds of transports, living in the villages, and etc. So basically, since the master studies, I was mostly occupied with these kind of surveys. And of course, the ecosystems in New Guinea are something which we already lost in North and Central America. The extents of the rainforest, which is not disturbed, are humongous, one of the largest still preserved forests on the planet. And it doesn't mean that the people don't live there. It's just that their agriculture usually / urban agriculture is not detrimental enough for the large conversions of the habitats, although this is quickly changing with the development of the plantations, etc. So the diversity of ants is extraordinary. So this is just an example of strumigenous species. These are small leaf-litter trapjaw ants living in one site. And as most of you know, dealing with the tropical diversity represents different challenges for the ecologists and taxonomists. So we developed standardized collecting protocols, which allowed comparative community sampling. And of course, all the specimen has to be processed by the standardized pipeline, which included photographing, sequencing, publishing in online databases, and sharing the distribution data with the major platforms as Antweb, Bolt, and other. So this is also the approach we are taking during the surveys of fauna in Mexico. Today, based at Lanaze, where we are, depending on the projects, we are using different molecular markers, for example, rat sequencing, universal microsatellites, Sanger sequencing, or recently UCEs, depending on the questions. But the idea is that all the specimens and the material is being processed in the standardized way, photographed, ideally barcoded, and then deposited in the way that is accessible to other researchers. And also, one of our main goals in those biodiversity surveys is to develop these long-term genetic resources for other systematic and biodiversity studies. And so coincidentally, actually, a big part of the specimens produced or collected in Mexico should be deposited at Instituto de Biología. So these long-term surveys of the fauna in New Guinea allowed us to start some studies about the community structure, about ecology of certain species. And today, although we are far from knowing the fauna well, we can already say something about the main patterns of the community structure and distribution of many of these species. So this is where the comparisons with the other areas and other islands becomes interesting. So some 5 years ago, we had the opportunity to survey Guadalcanal, which is this main island in the Salmon Island archipelago. And with the colleagues from South Pacific University and from AMNH, we sampled insects in the center of this island, which is now basically abandoned. And it was very interesting to compare our experience from the New Guinea with the species and with the community structure on those islands. So one of the striking things is that although the species are same to some extent or familiar, they are closely related species, the assemblage structure could differ considerably among these islands. So species which are, for example, rare in New Guinea are suddenly very abundant on some of these islands. They also change their ecology, such as our foraging patterns. There are also certain lineages which are not very species rich on the mainland, and then are suddenly very diverse in those island ecosystems. So in last year, we had the opportunity to also work in Bougainville, which is largest island of the Salmon Island archipelago in here, but politically it is part of New Guinea still. And for the last 200 years, there were only 5 records of ants from this island, and the situation for other insects is not much better. So we were able to survey some of those mountains on the island, which are mostly volcanic, but also the coastal region. That means we work in the villages or sometimes in the forest, in the camps. And these 2-month surveys allowed us to basically gather or expand the sampling and the data on ants from 5 records to about 3,000. So it's not a coincidence that this island hasn't received much research attention. Just 20 years ago, there was this harsh civil conflict ended on this island. The area was largely inaccessible, and of course, we can see a lot of consequences of this civil war until today, which makes it somewhat challenging social environment to work in. And just to mention that although we are primarily interested in research, working in Melanesia, almost anywhere where there are traditional communities, indigenous communities, is not only about the research. Important part of our activities, and it's actually the big proportion of it, are the discussions, training, and help with development of conservation or other environmental policies. We are also involved in some educational activities and also in some cultural activities, etc. So especially in Melanesia, every visit of the forest has to be very well discussed. All the activities has to be really in the very close coordination with the landowners and the traditional tribes, with the tribes. So another area at which we surveyed was this mountain in South Bougainville, which turned out to be quite interesting. And over 2 weeks, we were able to access one of the most pristine forests in the islands, which are still not affected by invasive species, unlike the lowland areas. So, and with the help of the local communities, we were able to find quite a large number of new species. So all these species are still undescribed, and they come from this one locality. And it was quite surprising for us to see some of these ants which didn't seem to be related to anything we knew from the region. Some of them are exhibiting also typical island syndromes that they are, for example, much larger than their relatives on the mainland. We also saw some interesting foraging activities. They are not as cryptic as the same as related species on the other islands, and so that is really was quite surprising and fascinating to see these large and quite common species which are totally unknown. So what is becoming more and more clear that there are like big shifts and changes in assemblage structure and ecological preferences among these islands. This is something which is not unexpected, which is not new, and for example, for botanists, that's something they are used to. But these things were not as much acknowledged in the biogeography research of or biogeographic comparisons of insects or invertebrates in general. So although we can have a species list for each of the islands, and the species list can overlap, that doesn't mean that the community is not quite distinct or different among these islands. We saw very large shifts in the species abundance, in range of habitat preferences, also in nesting preferences, and then there is a diversification of certain lineages which are rather poor on the mainland, but then they are suddenly very diverse on the islands. And there are also interesting phenomena. Although these islands are relatively, the fauna is several million years old, there is an apparent lack of arboreal ants on these islands, even if the same species or related species are arboreal in New Guinea or Australia. They are not doing this on those islands. So there are a set of questions which will be interesting to follow up. So one of these examples in the shift in habitat preferences is our favorite ant, Odontomechus similimus, which is this trapjaw ant, which is considered stage 1 species. And In New Guinea, it lives in the disturbed edge habitats on the coast, in the secondary forest, and it's rather cryptic and non-aggressive. However, when you come to Solomon Islands, this species constructs large nests. It lives in the primary forest, goes in the very deep into the islands, and it's quite conspicuous, and it's actually very aggressive. So now we would like to ask, from where did the ants come from in the South Pacific and Melanesia? There have been, since 60s, there have been several theories about the origin of Melanesian and South Pacific insects, often based on taxonomic comparisons, as here represented by work of Gressit. Also, Wilson made good predictions about how the species from the Oriental region are ancestors of New Guinean species, and these later expand to the Melanesia. But in some other groups, like birds or plants, we also are finding some evidence that species which are from Australia and New Guinea actually expanded back into the colonized Southeast Asia and then further, so as in those songbirds, for example. And then, of course, we have this pattern known from the human genetic studies, where we have this like sequence-like colonization of the islands in the jumping way from the Taiwan through Melanesia to Polynesia. So there was no big reason to think that this wouldn't be the case, although there were several cases where we know there are ants with slightly anomal ranges. For example, this is a Gnamptogenis, which is a diverse species in South and Central America, but it also is quite diverse in New Guinea and Borneo. But then there are other species, other genera like Adelomyrmex or Rogeria, which are mainly neotropical, and they have only a few species or one species in the Melanesia. So that seems like there was occasional long-distance dispersal. So our colleagues, several years ago, they analyzed the phylogeny of the Phaidoli, which is this big genus representing a big portion of the ants, and they reconstructed the evolutionary and biogeographic history, and find out that many of these species in Melanesia and in Australia are indeed of Asian origin. There was, however, interesting finding that species in Solomon Islands and Fiji, some of them, they came directly from Southeast Asia and not from New Guinea. And then many species also from living today in South Pacific and Melanesia are indeed of New Guinean origin. You can also see that there is a lack of this upstream colonization, basically from New Guinea and Pacific back to Southeast Asia. So then my former student Pavel Matos-Maravi, now our colleague at the Czech Academy of Science, reconstructed the phylogeny of the Prenolepis genus group, which represents several related genera. They are considered to be taxonomically somewhat challenging, as you can see on this photo. Each of these photos represents a different species. So we had to use molecular tools for species delimitation, and then we reconstructed the phylogeny, trying to ask what are the origins and patterns of diversification of Melanesian species, what is the sequence of colonization, and we were also trying to look if we can find some shifts in ecological preferences and geographical range expansions. So what Pavel did is to reconstruct the phylogeny of over 100 species from the region, and using our data, we were also able to reconstruct some of the habitat preferences and also the elevation range of some of these species. So again, this was an example where indeed this group originated mainly in Oriental region in Southeast Asia, and colonized the land which later became New Guinea about 20 million years ago. And there was a second wave of colonization from the same region about 5 million years ago. And in the meantime, some of these ancestral lineages also dispersed further into the Melanesian and South Pacific islands. So what was also interesting, however, was that we looked at the diversification rates, and we found that there was, in one lineage, there was accelerated diversification, which coincided with the shift in the ecological preferences towards the disturbed secondary habitats, and also with the geographic range expansion across the South Pacific. So here you can see the branch lengths scaled to the marginal probability of the rate shift events. So that's basically the diversification rate. And you can see that in this lineage, after the change from undisturbed to disturbed forests, there was really faster speciation rates, and also many of these species expanded into the South Pacific. And it's not probably coincidence that some members of these lineage are actually quite successful invasive species today. So again, here you can see how the diversification rates changed. They were pretty steady in most of the lineages, and here in this Nyland area clade 7, there about 5 million or 4 million years ago, they were suddenly very quick. We found that primary forest was the most preferred by the common ancestor in the Pacific clades, and highly disturbed and open environment was recovered only as an ancestor state in one clade. Yeah? So it seemed also that the colonization of the mountain areas in the South Pacific occurred only recently, less than 5 million years ago, and also it coincided with the shift into the disturbed environments. Yeah? Another study I just would like to mention briefly was Phylogeny of the Camponotus, led by our colleague Ron Klaus. And so that's a very diverse and common group of ants. And this was several lineages from Indo-Australia, where in the closely related lineages, we found quite distinct and different patterns of the biogeographic history. So each of these circles represents a different species, and these are reconstructed pathways of the colonizations among the islands. And so some of the species actually colonized New Guinea and Micronesia upstream from Australia, and some lineage colonized from Oriental islands towards Micronesia and then to New Guinea and Melanesian islands. But we also, in one small lineage, we found that the ancestors of these species, which today live in Australia and New Guinea, are actually now in Florida. So it seemed kind of rare, and it didn't seem to be artifact, but it was pretty clear from data. So it was kind of an interesting finding. And so then later, Pavel also reconstructed the phylogeny of the trapjaw ants, which are these charismatic ants of genus Odontomachus. They are very diverse in New Guinea and Borneo, but also there is a decent diversity in neotropics. And so they use mandibles not only for hunting, but also as a jumping maneuver for the escape from the attack. And so this is the sampling. And basically what we found is that there are three principal groups of these ants. The first one includes all or most of the species here in Southeast Asia and Oriental region. And then there are two groups, sister groups. One of them are New World species, and then all the New Guinean and Australian species. So it means that, and these are more closely related than the species here from Southeast Asia. So in other words, the species living here in Nearctics and Neotropics are more closely related to New Guinea and Melanesian species than species living here on the Oriental Islands. So that was quite unexpected. And there's maybe two explanations for that. And I would wait for the genomic data to confirm that. But it seems that clearly the Australian and Melanesian Ontomehus has a common ancestor with the New World species. So we also try to do some reconstructions of the habitat preferences and the dispersal rates between different types of habitats and different biogeographic regions. So here you can see the lineage of the Melanesian and Australian species. Each square is their geographic distribution, and this represents whether they live in the forest, in the primary forest, or in open and disturbed area. So, and here is the distribution of this species group. So you can see that majority of these species are really confined to New Guinea and surrounding islands, and there are only maybe 2 species which manage to expand across the larger areas. One of them is this species, which is actually quite successful, and it's because there was there is a shift in the very particular strategy where the species lives on the shorelines and in the mangroves and basically on the small atolls and etc. So this is quite unusual change in the habitat preferences and allowed the species to colonize large areas. But otherwise, you can see that these species are really often confined to rather smaller regions. And it's really interesting to see that in the Oriental region, we have these neighboring islands close to each other with a huge diversity of these species, and they come from, they have different evolutionary origins, and they are not really able to cross and establish themselves across the close islands. So that means that there are most likely other things taking place, like a interspecific competition, etc. So what we found in Ontomehus is also that the transition rate from the open habitats to forest interiors is much higher than the transitions from the forest interiors, from the primary forest, into the open secondary environments. So once the species lives in the primary forest, then it has quite a low probability to change and switch its habitat preferences and expand further. So the macroevolutionary rate dispersal is low for the species which live in the primary forest and forest interiors, and it's quite high for species which live in open habitats. So these are primarily the ancestors of those well-dispersing species across the islands. Okay, so and just another interesting thing in the study of Ontomehus was that if we look at this New World lineage, well within it is the Ontomehus similimus, which is one of the most common species in Indo-Pacific, which lives on the smaller and large islands, and it has clearly New World origin. And it seems that it expanded there about 5 million years ago. We didn't find any closely related species to it in the New World or populations, but it has clearly New World origin. So showing that these exchanges are probably more common than we thought they are. And again, just to mention, if we look outside of the ants, there has been many documented long-distance dispersals between the New World and the South Pacific, although this was often the situation for the plants or for flying invertebrates or for the things which are easily carried away by the wind. And I just wanted to remind this long debate about the origin of the sweet potatoes. This is culturally and nutritionally very important plant for for Melanesian South Pacific, and it's been hypothesized that it was either brought there from New World with the traditional trade, or another suggestion was that it came with the Spaniards and then got gradually with the local trade into the more remote areas. But according to the recent genetic studies, it seems that Ipomea actually expanded somewhere from Mexico by long distance dispersal several million years ago and established itself in the South Pacific Islands. And all these other archaeological evidence of the movements and so on are only secondarily. So, of course, these are some historic expansions and probably long distance dispersals of the lineages, sometimes unexpected. But in the past, in the recent years, we had also large number of expansions due to the human exchange and the commerce. So I just wanted to mention some which are related to Americas. One of these is invasive species Wasmannia auropunctata, little fire ant, which is native to Central and South America. It's rather inconspicuous and although can be pretty abundant in South and in south of Mexico and in some tropical Mexican forests. And in the 70s, it got carried into the South Pacific, where it's creating havoc in the local biodiversity. There have been it's been a huge problem in New Caledonia and Australia, and there are some successful treatments. However, in Solomon Islands and now in New Guinea, it got established and basically it's beyond control. So it's the set example of a really strong effect of the invasive species, which is practically lethal for the local biodiversity. That's was one of the saddest picture I saw last year. For example, in Bougainville, majority of the lowland forests along the coasts, they are colonized by this ant, and most of the native ant fauna is extinct. So there are large patches of the forest which is structurally well preserved, and all the native ant species are gone. And of course, it has also the cascade effect on other invertebrates and the vertebrates. And all these invasions are beyond control. And of course, it's also a nuisance for the agriculture and for the animals and for the kids. What is very common phenomena are these is this blindness, which is caused by the venom and the consequent fungal infections, and it's very easy to develop the allergies to this ant. So basically, it's a set example of invasion which is very strong, and practically there is no good solution for that. Another ant which also originated in Americas is this Solenopsis geminata, which is one of the most common ant in Mexico. You can find it on the side of the petrol stations. It's pretty aggressive. And so a few years ago, our colleague Dietrich Gotzek was able to basically date the expansion of this ant back to the practically almost exactly to the dates where the first Spanish galonas were leaving Acapulco for Manila, and as a present, they brought these ants into the Southeast Asia as in the soil, in the ballast of at the bottom of the ships. And this species was quite successful. So if you go today in almost any New Guinean village or places in Solomon Islands, you can be sure that you can find this ant below the houses, along the fireplaces, and in populated areas in general. Although luckily it doesn't seem that it's expanding into the primary or secondary forests. So just some conclusions about about this part. I just wanted to show you that we found some support for several predictions made by the Taksim Cycle using ants from the same area where this theory was developed. We also, it seems also that trade-dependent dispersal played important role in biogeographic history of several ant groups and in the Pacific. Unexpectedly, we have several lineages of ants which originated in the New World and not in the Southeast Asian rainforest, as it was considered to be the case for majority of the ant fauna in there. And there is a growing evidence for trans-Pacific long-distance dispersals, multiple ant lineages. We also saw that the human introductions of exotic species sometimes compensate for lower diversity, but they are often fatal for the local fauna. And so in the following 5 minutes, I just would like to show you briefly some of our ongoing research in Mexico. If anyone is interested in more information, please feel free to visit our website and hormigas.mx or contact us through the website or email. So one of the studies we've been focusing at during the past year was led by my colleague Madai Rosas Meija from the University of Tamaulipas. And Madai was working on assembling the database of the introduced ants in Mexico. So we basically put together all the records from the literature and through the collaborations of many Mexican colleagues. And now we have nearly 1,000 records for 40 species, which are not native to Mexico, distributed around 400 localities. All these data, we also did some basic analysis of the habitat preferences for these ants, and all these data will be available through the public domains. And the main reason for creating these resources is to share them and use them for some more systematic monitoring of the introduced species and for development of some prevention and control strategies. Related to that was also the project where we did the analysis of risk, analysis de riesgo, for the most important invasive ants in Mexico. And Madai developed very comprehensive reports and guides on the biology and prevention and the treatment of invasive species. It's probably on the best text, which is now available in Spanish. And we also created some materials which can be used for the identification of the invasive species, which can be used by the government agencies. And all this is accessible either through our website or through the CONABIO, which was sponsoring these projects. And so another ongoing research is the study by our PhD student Fatima Magdalena Sandoval Becerra, which is based at the University of Tamaulipas. And Fatima is trying to study the patterns of community phylogenetic structure in ants and chrysomeliid communities across the altitudinal gradient. So we have another altitudinal gradient being studied in the famous place with a long history of biological research. And this time we are trying to look more closely at the different evolutionary and ecological factors which determine the community structure. So we are sampling, we've been sampling across the 2000 meters of the altitudinal gradient, and with rather detailed and comprehensive sampling for each plot, and which allows us to also get the species densities and detailed knowledge of the community composition. So these are different methods which are being used per each plot, and so this is the sampling effort from the first year of sampling. So these are with several hundred samples of leaf litter, baits, or indirect collecting. So we should continue for one more year, and this should create one of the really comprehensive studies for the given region, with a lot of new material which can be which will be long-term preserved and used for follow ecological and genetic studies. And so when Fatima finishes her second year, that's going to be one of the best sampled locality for ants in Mexico, I suppose. Another project to mention briefly is the study of the ant assemblages across different environmental gradients across Baja California. Again, very important, well-known area of Mexico with a lot of conservation priorities. We are trying to evaluate different patterns of taxonomic, phylogenetic, and function diversity of ants, and also use comparative sampling across the whole peninsula, so we can also test some of the phylogenetic questions. And another important aspect is, again, building of the genetic resources for the other follow-up studies. So this project is led by our master student, Efrain Jesus Carrillo Vergara. And here on the left, it shows the sampled sites over the last 2 years. And here on the right, we have also the literature and databases data, so which are available for the peninsula. However, they are not often collected in the comparable way. So our idea is really to cover the majority of the habitats in some comparative sampling. And also important thing is that many of this material is basically not available in Mexico. However, all this new material should be, again, important part of the biodiversity collections for the country. And with that, I would like to thank you to our collaborators, sponsor, our students, and to everyone for your attention and the patience. And if there are any questions, we can try to answer them. Thank you very much.
Super. Thank you, Milan. Ok, sí, tenemos varias preguntas. Voy a darte unos minutos para que descanses de tu voz. Qué lástima que las hormigas siempre tienen nombres comunes muy trágicos, como Crazy Little Ants, and Fire Little Ants, bueno, las invasivas, ¿no? Invasoras. Ok, tenemos una pregunta por Carmen Huertas. ¿Qué tal compiten las hormigas exóticas con las nativas de México?
¿Cuánto compiten las hormigas exóticas con las nativas de México? How much do they compete with the... Sí, claro. ¿Cómo es la respuesta ecológica, no? Depende de la situación, depende de la especie. Obviamente tenemos algunos casos muy graves en el norte del país, que es la situación muy parecida como el sur de Estados Unidos, como Texas, Arizona, donde tenemos esta hormiga muy mala, Solenopsis invicta, y hay varios estudios de nuestros colegas del norte de México, de Veracruz, en la competición de estas especies. Creo que es principalmente el caso en estos ecosistemas más disturbados, ¿sí? Como de los pastizales y lugares que son afectados por la agricultura donde tenemos la diversidad de hormigas nativas es más baja. Entonces, allá tenemos un reemplazamiento. Una de las cosas que queríamos probar en Tamaulipas es ver si este efecto de Basmania auropunctata, esta hormiga de fuego, si hay un impacto para diversidad nativa. Pero sí, hay varios casos, pero afortunadamente no es como tan mal, aparte de algunos ejemplos, como la situación en las varias islas, ¿no?
Sí, sí, claro, yo vi cuando viví en Hawái como la Fire Little Ant ataca aves silvestres y algunos animales domésticos como los gatos, los deja ciegos y no sé si están establecidas en Hawái, pero sí es un peligro
Sí,
inminente.
exactamente. Sí, sí, es mucho más obvio en estos ecosistemas de islas. Pero obviamente teníamos algunos casos muy graves, no solamente los hormigas en las islas de Baja California o de Revillagigedo y estos tipos de lugares, ¿no?
Ok, otra pregunta por Rebeca Hernández Gutiérrez. Pregunta, ¿cómo se incluyeron las especies extintas en el análisis de FBD de odontomacos?
De FBD. Sí, es una buena pregunta y es poco para una charla, creo. Esto es un problema principal en los reconstruir y analizar, por ejemplo, el Taksim Cycle con los filogenias, ¿no? Porque es como algo típicamente muy difícil incluir las extinciones y especies extintas para que no tenemos evidencia para eso. Entonces, hay algunas escuelas de opiniones que prácticamente no podemos hacer estas pruebas, no podemos hacer estos análisis porque no consideramos especies extintas y estamos como muy preocupados por eso. Para algunos, para datación molecular y datación con los fósiles, sí incluimos especies extintas que teníamos, pero obviamente nunca podemos saber y cubrir las especies extintas que vivieron en estas islas. Y entonces es el problema metodológico que a veces intentamos saltar con varios, como otros tipos de análisis, pero sí es un problema.
O sea,
Pero,
la respuesta es que no se incluyeron en estos análisis las extintas.
no, porque si la especie es extinta no tenemos una evidencia, ¿no? Y aparentemente estos lugares no tienen los fósiles. Y incluimos varias especies extintas en la análisis de Odontomechus, pero era más por la datación y por la... por ejemplo, hay algunos estudios como más globales donde podemos contar o tener unos modelos cuántas especies fueron extintas por la tasa de especiación, pero yo soy un poco más conservador en estos métodos, entonces sí, eso es un problema, obviamente.
Ok, otra pregunta. Karen Beatriz Hernández escribe pregunta, bueno, gracias por tu plática. ¿Cuáles son los retos que se enfrentan para describir la variación morfológica de las especies descubiertas en el Indo-Pacífico?
¿Cuáles son los retos que enfrentan para describir la variación morfológica de las especies descubiertas en el Pacífico? Significa
If there are any challenges to describe the morphological variation in the...
sí, claro. Principalmente la situación es muy parecida como tenemos aquí en México, ¿no? Con los geografía muy compleja y diversa, tenemos varias subpoblaciones, una variación morfológica larga, pero solamente es la cuestión de esfuerzo de muestreo, suficiente muestreo y obtener material suficiente para comparar esta variación. Varios de estos estudios juntamos material para casi 15 años, solamente de nuestros estudios, y utilizamos mucho material de museos también, incluyendo la secuenciación, pero prácticamente hoy los métodos que utilizamos son la secuenciación masiva con RADseq o barcodes para combinar esta variación morfológica con variación molecular y llegar a un punto donde podemos utilizar algunas unidades que son comparables, ¿no? Porque depende de la escuela y historia y eso, cada persona o cada instituto se va a tener poco diferente opinión que son las especies, cuál es la variación morfológica y cuáles son diferentes especies. Eso se cambia mucho. Entonces, ¿qué estamos intentando? Es tener algunas unidades que podemos comparar y que después, cuando se cambian los métodos o los conceptos de especies, que alguien puede revisitar y corregir nuestros datos, ¿no?
Ok, Rebeca, la chica que hizo la pregunta anterior sobre la inclusión de las especies extintas en los análisis, menciona que ella hizo su pregunta porque habían tips que no llegaban al presente en la filogenia. Esto no lo entiendo muy bien. No sé si me perdí de esta parte en tu charla, pero
sí, claro. Sí, eso fueron los fósiles que creo que teníamos para datación de la filogenia, ¿no? Utilizamos muchos de los fósiles y recomiendo, el artículo es de Molecular Ecology hace 2 años, creo, entonces, y es disponible en nuestra página. Es un artículo un poco complejo, entonces yo simplifico algunos, hay como mucho más aspectos de este artículo, pero sí, incluimos fósiles para eso. Okay. Pero no para estudiar el Taksim Cycle con las especies extintas.
Okay. William Rincón dice, ¿registraste nuevos comportamientos entre los gremios que investigaste? ¿Puede un género perteneciente a un gremio cambiar a otro gremio? Between guilds, if there are movements between guilds.
Dentro de las islas, ¿no? Entiendo. En estos gremios, registran los comportamientos entre los gremios que investigaste. Sí, eso es. No tenemos una documentación tan buena para eso porque es un compromiso, ¿no? De entre estos lugares son muy diversos y prácticamente es poco difícil mostrar algo de una manera repetitiva y para seguir con mismas comunidades en largo plazo, ¿no? Es muy caro, necesita mucho esfuerzo y a veces las prioridades son que queremos enfocarse a alguna otra área que no fue tan reconocida, pero particularmente entre las hormigas, en como más largo plazo, creo que puede tener algunos como shifts en los comportamientos, ¿no? Hay como varios estudios que son estudios de interacciones entre las hormigas y utilizamos, por ejemplo, trampas de cebo para evaluar la, ¿cómo se dice?, jerarquía de dominancia. Y eso son cosas que son relativas, porque depende mucho de la composición de un gremio o asamblaje particular, cómo se comportan varias especies. Y si llega otra especie que es poco más dominante o con diferente conducta, eso puede tener una consecuencia en la conducta de los otros también.
Ok, hay una pregunta por Ana Santa Cruz. ¿El mayor éxito de un linaje en la isla en comparación al continente dependería más de la ventaja del colonizador o del subyacente genético del linaje? Por ejemplo, variación genética escondida que se dispara
desde eso es interesante. Yo quiero saber esta respuesta porque es una pregunta como
grande.
Como grande un poco y también puedo decir, depende de la situación, ¿no? Entonces, tenemos varios escenarios y a veces si no esas son cosas que se ocurren a largo plazo, ¿no? Entonces, a veces es difícil reconstruir qué pasó con estos ancestores, ¿no? Y la situación con introducciones recientes o especies invasoras es muy diferente. Entonces, creo que puede ser ambos, depende del caso.
Sí.
¿Sí?
Ok, tenemos otra pregunta.
Perdón, no puedo responder mejor.
Do you think that the dispersal patterns of ant species in the Malaysian region could be the same for the tropical islands of America? How could this process be explained?
Hola Fátima, gracias por la pregunta. Sí, claro, eventualmente estamos imaginando que estos patrones son patrones un poco más generales, ¿no? Estamos oscilando de entre como historias de vida de algunos lineajes, pero también queremos llegar en los patrones más generales que que está repitiendo en los varios lugares e islas. Entonces, para que podamos utilizarlos para hacer predicciones más generales. Entonces, sí, pero como podemos ver, también depende mucho de las áreas de origen, ¿no? Por ejemplo, de las islas, de estos archipiélagos largos que tiene México, tiene una geografía e historia muy diferente, no son tan diversos, porque depende de su clima, depende de los corrientes. Entonces, a veces es muy idiosincrático, ¿no? Entonces, depende mucho del lugar. El avantaje de estos lugares en Indo-Pacífico es que tenemos muchas islas con condiciones más o menos similares, parecidas, y allá podemos ver la evolución en los mismos o especies muy relacionadas con condiciones prácticamente iguales, pero se termina en los diferentes resultados, ¿no? Entonces, sí, pero para eso también recomiendo mucho del libro de Jonathan Lossos y de los contingency y randomness de la colonización de las islas en Caribe y Mesoamérica, ¿no? Y todas estas cosas son bien descritas allá.
Ok, muchas gracias, Milan. Estas son todas las preguntas que tenemos. Gracias a todos los que se sintonizaron hoy con el seminario. Antes de despedirnos, quiero recordarles que cuando regresemos de vacaciones tenemos otro seminario el martes 28 de julio. El seminario será por la profesora María Alice Neves de la Universidad Federal de Santa Catarina, Brasil. Ella nos va a hablar de Fungal Diversity from Restinga, an Endangered Ecosystem in Atlantic Forest en Brasil. Muchas gracias de nuevo, Milan. Muy interesante tu charla.
Muchas gracias a todos. Gracias por la atención y mucho gusto.
Que tengan todos buenas vacaciones y cuídense.
Gracias también. Hasta luego.